Good morning. We're going to get going. All right. It's my pleasure to welcome you to the 35th anniversary Hans Fischer Lectureship here at the Department of Nutritional Sciences. My remarks will be brief, but I did want to point out that this is our 60th anniversary as a department. We were born as the Department of Nutrition in 1966. And in celebration of this, starting in the fall semester, we'll have plans to be celebrating the department, including we're going to be announcing at various times 60 different things, exciting things that the Department of Nutrition over the years has done. And so look forward to that celebration in the fall. But what we have today first is a big treat, and that is Hans Fischer, who was our founding chair in 1966, is going to give us some opening remarks on Zoom. And so hopefully everything's working right. Take it away, Hans.
Thank you. I'm absolutely amazed that my connection to Rutgers started in 1945 when I came to the university, which was supposedly Rutgers University. We were put in a bus, and the bus took us to a military camp where we each had one bed in a large hall, because the university had been a small college, was called Queen's College at the time, and they didn't have enough dormitories at all. That was only when it really became Rutgers University and part of the state university educational system. So I remember being out there and having one bed and being transported back and forth in the morning to the so-called campus, and at night back to that big military camp where we all had just one bed. So that was my introduction to Rutgers University. And then, as you have heard, and as you undoubtedly know, in 1966, it became the Nutritional Science Group became a separate department. I still remember when the president gave a talk to the American Dietetic Association in Philadelphia, and he announced that the university was going to start a department of nutritional sciences. And no one had mentioned this to anybody from the precursor of the Department of Nutritional Sciences. In any case, there was a group of nutritionists from various departments, and they elected me to become the chair of that department. And I spent 56 years at Rutgers and had a whole bunch of experiences most of them good, but also some bad ones. But that's what life is all about. And it was certainly a most interesting enterprise for me, and I would imagine for those with whom I interacted. So I look forward, as I hope you are, to the lecture now, and I'm very happy that these lectures have been going on for a long time. By the way, they were basically started because all departments at Rutgers were reviewed every 5 or 6 years, and including this department was reviewed, and the chairman was a biochemist from the University of Wisconsin, and he and his committee made the recommendation that the department at the university would do a good deed if they named a lecture after my name and brought outstanding researchers to the university to give a major lecture and have an opportunity for students, graduate students and even undergraduates, to meet with the speaker and get an opportunity to learn the extent and importance of nutritional science. So I'm very happy that we have a speaker today, and I look forward to listening to him as well. All the best.
Thank you, Hans. It's indeed a great honor to be able to put on this this lecture series, and we thank Hans for his remarks, and we're so glad he could join us today. Next on our list, I want to introduce to you Executive Dean of the School of Environmental and Biological Sciences, Laura Lawson.
Good morning, everybody.
Morning.
Come on, energy. I know. Woo! There we go. It's a delight to be here, and I really appreciate this opportunity to welcome this group. When I was preparing, I look in my files and I see every year the welcome that I write up and never look at, right? And it's like a diary of your department, of the people that have been involved in organizing it, the guests that we bring, and it's just a really wonderful tradition that the Department of Nutritional Sciences has. So I just want to acknowledge that. I also want to just mention, I mean, what a treasure to have Hans Fischer here today with you. So his connection, if I heard him right, say 1946, that's an 80-year member of our community who is still giving to Rutgers. That gives me chills just thinking about it. So thank you for being here, Dr. Fischer, and thank you for your continuing support of the department. I can't think of another department that has that kind of connection to its founding. It's very exciting, 60 years. That's going to be easy for me to remember because that's about my age, So that gives me some perspective to think about everything that has happened in 60 years. And if you think about nutritional sciences, how different it is today than it was in 1966. And I can't wait to hear the 60 things that have been discovered or done in the department. But we're here today to welcome our guest lecturer, Dr. Greenberg, who is coming today to talk about his work. Dr. Greenberg has made many discoveries, and I'm sure Dr. Shapsis will give a much greater detailed information. But what's really important is that we're continuing the conversation about the relationship between nutrition and obesity, and the different discoveries that have happened to better inform us about how to address this chronic challenge in society today, and to go deeper dive into what it means in terms of better care, better planning, better ways to address long-term health. I'm sure all of us have direct connection with some of the health consequences of obesity through our family and friends, and so I just want to I give my gratitude for the work that you are doing, because it is so important to humans as we move forward. I also want to take this opportunity to thank those that organized this event, the faculty, staff, and students of nutritional sciences. Life is incredibly demanding. It doesn't seem to be slowing down. It's super important that we all get together every now and then as a community and have this opportunity to learn together, and then take that as the beginning of conversations that keep going. And it's that stuff that really catalyzes us, gives us energy, keeps us moving. Your emails, they'll just keep coming, right? All of those things that you have to do back in your office, those will still be there. But this is what we're supposed to be doing. So I just want to thank you all for being here today. Thank you to the team in nutritional sciences for putting this together. It's never easy. The planning for next year started yesterday? Probably, yeah. So with that, I just want to thank you all for being here. Have a wonderful time, and enjoy your time here at Rutgers.
Thank you, Dean Lawson. In that spirit of thanking the people responsible, first of all, Christina Duffy, who arranges almost all of this, thank you as always. And then our seminar committee, chaired by Harini Sampath, including also Malcolm Watford, Sue Shapsis, and Joe Dixon. All right, now it's my pleasure to introduce to you Sue Shapsis, professor in the Department of Nutritional Sciences, who's going to give the speaker introduction.
No, I don't want my picture. Okay. Did you guys hear what I said? Yes. I wanted the speaker's picture up there, not mine. So it's a pleasure to introduce you, Dr. Greenberg. He leads the metabolism and basic biology of aging program at the Jean Mayer USDA Human Nutrition Research Center on Aging. It's a long one, but it's a very well-known one that we may or may not say the full words. He's also the co-director of the Boston Nutrition Obesity Research Center. So there's only a few around the country that are funded. Indeed, we have one in New York, but the Boston one is very active, and we know him from that, and it takes a lot of work to run something like that. So thank you for your co-directing of that one. And then you are also a professor of medicine at Robert and Veronica Atkins professor at Tufts University School of Medicine. Let me just say a few words about his background, in that he got his MD from NYU, and then he went on to training for many, many years at NIH. And I thought that was very key to his interest in research, and it really made a difference. He was in the diabetes division. He was also in basic sciences divisions and cellular and developmental biology. He has an interesting array of grants over his history, lots of R01s. He also has T32s. That means he's involved with a lot of training. And then, of course, the P30 that funds the NORC, the Nutrition Obesity Research Center. So this is a comprehensive person who engages in science and education in a variety of ways. Let me go to what he studies. It's adipose tissue biology and metabolic disease, and he's credited with co-discovering perilipin. We had one of the other researchers in Perry Lippen in this very department for quite a long time, Don Brasamley. And it's amazing that you were involved with the discovery of that. It is involved with fat storage and mobilization in cells, and it contributed to obesity and insulin resistance understanding. No more with all the information about him, but he has many awards, and one was from the Obesity Center that I noticed, and then the other one was last year at American Society of Nutrition. And with that, I'm going to tell you that's the reason that I met Dr. Greenberg by chance at that moment. We were standing in line in an awards photo line, and he started telling me about his recent paper, and it was really interesting on hyperinsulinemia, adipocyte, TPH2, serotonin, and how it was involved with metabolic dysfunction and obesity. So the two takeaways are that keep doing interesting work, and then make sure that you can explain it in a way that sticks with your colleagues to remember later in the year, hey, I really heard something interesting from my colleague this year. And here you are today. But the first time I met Dr. Greenberg was via Susan Fried, and I was at Maryland. And she warned me, when you meet Andy, either look way up or look at his belly button. And I opted to look way up, as you would imagine. Okay, anyway, please join me in welcoming Dr. Greenberg and his topic here. Okay, thank you.
So thank you. I'm honored to have heard Dr. Fisher and his contributions. One of my favorite slides for science is a Dr. Seuss slide. And in this slide, I should have used it today, is a bunch of turtles standing on the shoulders of each other. And the concept is that what we do today is built on the shoulders of other people. And all my research is built on the shoulders of my people in my lab because I don't go and do it. So I've been very fortunate to have great colleagues here, Judy, Susan Fried, and others, and people in my lab. So now I think after all those speeches, I have 5 minutes left. So, all right. So I will say I'm a little embarrassed by all the hoopla and honor. It's very nice. I just love what I do, and I'm fortunate to do it. So a little bit of background further. So I went to NYU Medical School. I was a medical resident at Penn. I met my wife on a blind date while I was there. The best thing I ever did. And I was going to go work with Brown and Goldstein on, who many of you know won a Nobel Prize. This is before I won it. But my wife said, my future wife said, we're not going to Dallas. So I went to NIH. And basically at NIH, I spent 9 years. I actually quit for a while because I didn't think things were going well. And I was going to take a private practice job in Philadelphia. But with Dean Londos, we discovered this highly phosphoric protein. He said you could clone it. And my wife, who is super great, said, even though she's from Philly, she said, why don't we just stay and you give it a try? You seem unhappy. And we're fortunate that we found Perry Lippen because it put my kids through college. And while that started me going, and I will talk about a number of different things today, which will cover different areas. So we've kind of metastasized in our scientific research. So my only potential conflict is, and you'll see why, is I have some funding or interactions. I really a grant from Novo Nordisk. So I put the next slide in. This is a Susan Fried slide, who is my honored and one of my best friends and colleagues. In 2026, we probably would want some women Susan in this, but it's all right. All right. So I work on obesity. Why do I work on this? So I'm 6'5". My mother was 5'2". My parents are very poor. And I said, how did I get so tall? And they said, oh, it's because we were poor. We couldn't afford food, so we gave you food. My mother was a great cook. I told her if she had made the plates a couple inches larger, I would have been a professional basketball player. Unfortunately, it never happened. But the other thing is my mom, unfortunately, became obese, developed diabetes, and had a stroke and died from it. So it got me interested in obesity and genetics. So that's how I came to be working on this. All right. So let me stand here maybe. All right. So there's two real projects here, and they're very diverse. So I never thought I'd be working on the intestine in 1,000 years. But we started working on these ACSL proteins. And this is because of a colleague, Rosalind Coleman from UNC, and Doug Mashik. As you'll see, ACSL5 is very highly expressed in the intestine. And I was fortunate to have a graduate student done a master's in intestinal biology. And I said, well, let's do this, but I think we'll end up with diarrhea, but you'll see. So, so the good news is if you have students, they can make these animated slides, which I can't do. All right, so, you know, this is no kind of high caloric diet. You gain weight, it enters your stomach. And, wow. Oh, we're going the wrong way. I don't know how to drive this. Okay, so it gets absorbed in the intestine, and the regulation of food intake is complex. It's the brain, endocrine signaling. This is the small intestine. There's the blood vessels. You know, the actual, this is not totally correct. Of course, the lipids, et cetera, end up in the lymph. And actually, the enteroendocrine hormones end up in the lymph. They don't go right into the blood. That's been shown by a colleague in Ohio. But one of the important things here is this. So this is the enteroendocrine cell. You know, when I started research, I was working on GLP-1, and I submitted a grant, I don't know, 6 years ago. And the first reviewer said GLP-1 is not important in humans. So it shows you what life is going. Honest to God. So, you know, you can never stop. But even my family, who have nothing to do with research, know what GLP-1 is now. You know, you go, oh yeah, GLP-1, sure, no problem. Anyway, so GLP-1 is secreted from enteroendocrine cells, which are specialized cells. They are regulated by glucose and lipids, amongst other things. When they bind, they cause secretion. The glucose and lipids are actually on the basolateral side. This is not correct. And then you secrete like GLP-1 and CCK. CCK actually goes in the blood, causes, in addition to regular food intake, it causes contraction of the gallbladder, it spills bile acid. GLP-1 acts, initially was discovered because of its importance increasing insulin secretion. And then when they did these drugs, they were agonists, they found out indeed they regulated body fat. Okay. So how do ACSLs function? So what they do is when fatty acids enter the cell, either by synthesis from acetate, for example, or diffuse in, the long-chain acyl-CoAs, acyl-CoA synthetase, activate or put a CoA group on the fatty acid. Now, what's critical about that is if they don't put the CoA on the fatty acids can diffuse in and out of the cell. And we have examples in mice and in humans that if you don't put the CoA on, and then you look, the fatty acids are gone. For example, arachidonic acid, we showed in macrophages that are ACSL4 depleted, they have 80% reduction in arachidonic acid, and they can't make arachidonic acid metabolites. That's another project. Okay, so this is the concept. So just to introduce you who don't know Dr. Storch's great physiology work, in the intestine, excuse me, so triglycerides broke down to fatty acids, 2-monoacylglycerol, and they entered the enterocyte, really mostly jejunal ectocytes, and then they have to be reesterified into triglyceride or cholesterol ester. So here's a monoacylglycerol, and you need to form, with the fatty acid, you need to form a fatty acyl-CoA, and they, in the actions of MGAT, form diacylglycerol. You need another fatty acyl-CoA. With DGAT, you get triglyceride. The triglyceride ultimately gets packaged into chylomicron and secreted in the blood. So it turns out that ACSLs are very important in fat absorption. So what about ACSL5? So these are two different papers. The bottom is from my lab, the top is from another lab. And they look at the relative expression of ACSL5. It's a little cut off, I apologize. But on the left, you can see ACSL5 is the most prominent ACSL. This is not so important, FATP4. But on the bottom is from our lab. And you can see this is the tissue distribution of ACSL5. There's some in brown fat, some in liver, a lot in small intestine. Now, these are low-fat fed mice. Obviously, when you feed a high-fat diet, you can change the expression levels. So I said to my graduate student, let's knock it out in the intestine. So we mated the ACSL5 flox mice we generated to mice that had what's called an inducible villain-Cree. So villain-Cree most specifically expresses intestinal epithelial cells. And if you add tamoxifen to the mice, we developed a way to do it orally. You can generate either the knockout mice or you have control mice. I said, okay, do this. This is going to be a short paper. The mice are going to malabsorb the fat and we'll be done, but you'll get a nice short paper. Ha! All right. So here's just showing that we have excellent knockout. This is the gene expression. And on the top is the protein, which still counts in life. And the liver, you can see, is no effect. It's very specific. So to my surprise, we fed him a high-fat diet. Not surprised. And you can see that within the first 2 weeks, so you can see chow has no effect, which is the white and gray on body weight or body fat. The knockout is red, and you can see even during the first 2 weeks, there's significant protection against diet-induced obesity. It's just remarkable. I said, okay, that's because they're spilling the fat into the intestine. We'll get to that in a second. And this is more long-term. Top is low fat, here on the left. Bottom right, long-term high-fat feeding, it works. And, you know, adipose tissue and liver weights on a high-fat diet are surrogate markers of fat accumulation. You can see that in EWAT, the fat mass is down, IWAT is down, and in BAT. Liver weight, so less triglyceride, so that's good. We injected, we did a glucose tolerance, insulin tolerance test, just standard tests. Not surprising if they're thinner, that would be improved. And fasting glucose is improved on the above. A, B, fasting insulin is decreased, consistent with improvement sensitivity. If you have any questions, just raise your hand. So when I was in college, my first course in chemistry was with the most intimidating professor I ever had. He went to Harvard, got a PhD from MIT, and pitched for the Chicago Cubs. And he was super smart. And he said in the first day, in this class, it's acceptable to be stupid, but not ignorant. If you have a question, you know, just ask it. So I always tell that to people in my lab.
So why the oral delivery of tamoxifen? Do you find that makes any difference?
We could use less of it. We use a very small amount. And there are problems with the IP because the IP can actually sometimes, Vanderbilt researchers showed that you can get a little nidus and lasts forever. So this way it goes in and out. And we need very little. It works very, very well. To John's credit, he was really good. You know, I was very fortunate. Anyway, so C, this is from my paper. I apologize that it's from paper. But C and D is insulin tolerance. You can see the area of the curve is improved, even percentage-wise, and the glucose tolerance. So that's good. All right. So what's the mechanism? All right. This is the nitty-gritty. So there was no difference in energy expenditure. Anything was slightly reduced. Well, okay, maybe they're malabsorbed and they've got a problem. So we measured their fecal lipid output, which is the amount of lipid in there with Kim Booman, and there was no increase. This was a surprise to me, I can tell you that. And then, I don't know, this is like magically moving forward for me. I don't know, God's on my side. Anyway, but what was most prominent was the reduction in food intake. And so that was surprising and meant I had to go read 1,000 other papers to figure out what the hell's going on in a new area. Oh, this is really wild here. Okay. So I'll put this down. So we made a simple study design delineate the relative importance of food intake, kind of an old-fashioned way. So we fed the flox mice the same amount of food as the knockout mice that they ate, okay? So I couldn't do this, but my student volunteered to do this because he wanted to do it and went in every day, or actually every 2-3 days. And so the concept is they have the same food intake, the knockout and the flox pair fed, but the flox control ad libitum was not. So it's really pretty simple. So he achieved this, and you can see the nice colors and nice diagram. On the left is the wild-type mouse, not pair-fed, and its average daily food intake. But the knockout and the pair-fed have similar. How does this happen? Maybe it's timed. And you can see that they have the similar body weight, the knockout and the pair-fed. And if you took out the fat pads, like I told you, you get a surrogate marker of fat accumulation, and you can see it's reduced significantly in the pair-fed knockout, in inguinal fat, epididymal fat, pretty close, mesenteric, and brown. So, get to the, that's cheating. All right, close your eyes.
Maybe I could do it and you just say next slide.
No, yeah, that's okay. I think it might be timed, actually. I'm not sure. Not timed?
No, it could be.
What?
It could be time if it's going ahead.
You know, these students. Anyway, thank God. Okay, well, I should say that in 2026, but very thankful to have them. Okay, so what's going on here? So we got clues from these experiments. So the first thing, we measured villus height, and I can discuss that later. The villus height, you know, these intestinal villi go up, and they had surface area. It was actually increased in the knockout, which I think allows for more surface area for the absorption of the dietary fat. Okay. Then we did what's called postprandial triglyceride studies. On the left, we just fed mice triglyceride and measured the rate of appearance of triglyceride in the blood. And in the right, we added taloxifol, which inhibits LPL, Susan Fried's enzyme, and we can see that it's still decreased. So the rate of appearance of triglyceride after absorption of fat is decreased. That suggested something to us. And so what we did is we took radio-labeled trialine, mixed it with fat, and then measured the amount of radioactive and lipid in gastric intestinal mucosa. And what you can see is that after the bolus of fat in the knockout, there's much more fat retained in the stomach, the gastric, and there's less in the intestinal mucosa. So that's slowing the rate of gastric emptying, which was a big clue. Again, the fecal fat was the same. So that led us to ask, is there an enteroendocrine hormone, which is, you know, with the famous ileal brake where you secrete them. You probably all know about it. You probably know from the Today Show that GLP-1 slows the rate of gastric emptying. And so maybe that's it. So indeed, we did various measures in our knockout. And on the upper left is we gavaged the mice with olive oil. Olive oil is a very potent stimulus of GLP-1. Okay, you still passed the course, don't worry. So you can see that active GLP-1 is increased after the knockout. This is right away. Then we did the fasting refed with a high-fat diet. Again, it's much higher. And on the right is after high-fat feeding. After high-fat feeding, the amount of GLP-1 secreted on the left is in the gray is dramatically blunted, but maintained in the knockout. Okay? All right. So then we contacted this group in Indiana, a guy who made drugs against GLP-1, and we got a GLP-1 long-acting agonist. And so in the study, indeed, we tested this in mice. We injected the agonist into mice, and on the left is just saline and GLP-1. So no difference. But on the right, well, we took so there's no difference in the flox. But in the knockout, with saline, you see the red. But when we added the antagonist, now this we didn't titrate it, there was increased food intake consistent with some role for GLP-1 in regulating food intake, okay? Then the reviewers said, oh, what about other proteins? And we found that PYY was also increased. So it's not a single protein. Okay. And then I presented to you in the beginning that ACSL5 trap fatty acids in the cell. So we had a very simple hypothesis, which is pretty much true, is that if you had reductions, so ACSL5 is 80% of the ACSL activity, you eat a fat diet, the fatty acids don't immediately get trapped into the jejunal cells. They get to float in and out and bounce around. And when they float in around, they have greater likelihood or greater time to bind to enteroendocrine and cause increased GLP-1 secretion. And you can see that this is ileal fatty acids are higher in the ileum, in the lumen of the ileum of the jejunum, consistent with one hypothesis. So then, you know, the thesis committee people always have ideas. So one of them was a neuroscientist, says, hey, do they have a preference for different kinds of food? So in the cages, we put low-fat, high-fat diet with the flock, the knockout mice. And so the knockout mice, high-fat, ate less. Okay. The relative high-fat diet intake was slightly less. But what was interesting is that the knockout greatly preferred the low-fat diet. So there's some food preference. Don't ask me why, how in the brain it does it, but it's an interesting fact. So, and then I wanted to show you this slide. So this is the pair feeding. And you can see the pair feeding doesn't affect the GLP-1 levels like they do in the knockout. The right's the knockout. The pair fed's the purple, but when you measure serum active, it doesn't account for that. The pair feeding just controls it, but this knockout still makes more GLP-1. So like this, I keep forgetting. So in summary, mice with intestinal ACSL5 deficiency are protected against diet by increasing certain levels of endocannabinoid hormones, at least in part. Okay? And we have no change in energy metabolism or physical activity. So we published this paper, and then there's a company called Lexicon Pharmaceuticals in Texas, and they make knockout mice. And they had noticed that ACSL5 knockouts, which we had published already actually, were protected against diet-induced obesity. So they made a drug. So we published this paper, and they said, gee, that's great. We now know the mechanism by which this drug is working. Well, that was nice. So they went ahead, and they sold the drug to Novo Nordisk for tens of millions of dollars. Now, this is after my postdoc said every day, why aren't we making a drug? I said, we don't really make drugs, but we could have, I guess. So on the bottom, you can see it's discovered that it's an ACSL5 inhibitor of its activity, and is now in phase 1 trials in Novo Nordisk. So I don't get any money for this, but I have a study looking at ACSL5 activity in human small intestinal organoids. Okay, that's where that comes from. Unfortunately, my wife was not happy with this, but there's nothing we can do. All right, maybe next time. Okay, so we're going to switch gears, something for everyone today. I'm going to talk about I don't know why our projects get so complicated, but this is Brian Park's project. I'm not sure I wrote his name in the end, but he did the bulk of the work. And this is about a adipocyte enzyme, TPH2. So on the top, I had a graduate student in my lab who we were doing ACSL4 knockout, and we didn't know why they're protecting against diet. So I said, why don't we just do RNA-seq of high-fat diet adipocytes and knockout from the ACSL4, which are reduced body fat. I said, nothing is going to be good about it. You know, we won't get anything, but go ahead. This is before everyone in the world was doing it. And so we found a couple hundred genes upregulated and downregulated. And I said, wow, we were considering putting a statue of her in the lab because of our contributions. But she found the most downregulated gene in the knockout was TPH2, 50-fold in adipose isolated adipocytes, which I said, can't be that we have stumbled on such a thing, not us. And the literature has other papers that show that it's downregulated, it's upregulated in obesity. Here's the bottom: bat, gonadal, lutein, mesenteric. So, okay. So we decided to go after it. So what does TPH2 do? So this is nutrition. You all know what tryptophan is, I hope. And tryptophan gets converted by TPH1 or 2 into 5-hydroxytryptophan, and then ultimately into serotonin, and it can be broken down. Okay? TPH2 is almost thought to be predominantly in the brain. TPH1 is in the periphery. So here is what I just said. My students make better slides than I. So central is TPH2 in the brain, and also it's thought that maybe it can regulate intestinal motility. Serotonin can regulate, it'll slow gut motility. Keep that in the back pocket. And 90% of peripheral serotonin is TPH1. There's a guy at Columbia, I blanked his name, Susan may know. He does a lot of intestinal stuff, and he showed that intestinal secretes a lot of serotonin. Okay. All right, well, we got that. So these are various functions. It turns out that 5H, here, so cool. I mean, you know, like the movies. We should have IMAX screens, right? So TPH1 is increased, its expression in obesity. And it does decrease thermogenesis. And may affect lipid accumulation, affect white adipocytes. But the peripheral role, TPH1 had no effect if you're knockout. You knocked out, no effect on circulating serotonin levels, which is critical. However, the peripheral role of TPH2 had not been investigated. So we decided to kind of follow this up. So this is high-fat diet. This is the amount of serotonin, 5-HT, in adipose tissue of chow and high-fat fats. Obviously increased. You don't, you know, even the plasma 5-HT was also increased. Whoa, that's kind of interesting. We thought probably intestine, right? And in isolated adipocytes, like, you know, like real scientists, there was a little increase in TPH2, but a big increase of TPH2. Little increase, big increase, also in brown. And we did protein blots that confirmed TPH2 expression. This guy used up all the money in my lab. So we collaborated with Mike Jensen. Mike is a human researcher, because I like to go back and forth. And I've been fortunate, a lot of things we found. Perilipids in humans has big effects. Okay. So Mike was kind enough to send us adipocytes from lean and obese, which is here on the bottom. And you can see TPH2 is increased in obese adipocytes of humans. This is subcutaneous, okay? And then Mike is like, he's a clinical endocrinologist. Oh, let's start correlating everything. So we saw that the expression of TPH2 correlated with the liver enzymes, suggesting, you know, fatty liver didn't have much effect with cholesterol, but was negatively correlated with HDL. So seems important. The good news, it's in human adipocytes. So I felt better about it. So we have our mice, and we designed some very simple, we use adipocreme mice, knockout and flocks, the chow high-fat diet, and away we went. Here's the diet if you want. So the first thing you'll see on the chow diet, high-fat diet, they have the same body weight, the same fat mass, and same lean mass. Okay? hold on tight. And then we measured tissue weights. It was reduced in EWAT, also in BAT, not shown here. Oh, here's BAT weight. It's reduced. So even though there's no change in whole body fat, there was some change in the depot weights. Okay. And to our surprise, up on the left, you can see that there's decreased plasma serotonin. So adipocytes were secreting the serotonin. So TPH2 generated adipocytes generate serotonin not only acting locally, but secreting in the blood. And it would seem to be regulated by fasting, also slightly decreased by fasting. We'll come back to that. And it was, and the insulin was, oh, this is glucose. So they have lower glucose to knock out, lower insulin, and they had somewhat improved GTT and ITT. He injected insulin because he had to use up more of my money, and did a Western blot for phospho-AKT. It showed that it increases phospho-AKT in liver, muscle, and adipose. So he's a very careful guy, in all honesty. Very good scientist now at Joslin, doing a postdoc. And, no surprise, so the knockout seemed to have less liver fat, which is right here. This is a picture, but measured. And the enzymes of liver damage, which is usually consistent with liver fat, were decreased. And you have to read this. And then this is the relative expression of the enzymes. And what about adipocytes, our favorite? So they had decreased adipocyte size. They had decreased crown formation. I don't know. The crown is something we found as the adipocytes get bigger, in both humans and mice, they tend to die. And when they die, they leave their remnant lipid droplet behind, and macrophages come in and surround it and sequester it, but they also get activated. So it's part of the inflammatory cause. Actually, this paper, our first paper, is over 2,000 references. And you can see here, these are pretty easy to see. These are crown-like structures. And there's a lot less here, okay? Those are just macrophages. And actually other cells as well. And there's some decreased lipogenic genes in the adipocytes. Okay. She can open the door? Okay. And so CLS, crown-like structure, you can see are greatly reduced. All right. What about energy? Well, they have the same body weight. What about energy? Whoa. They have increased energy expenditure, which was a surprise. Of course, that made it more complicated and a little bit more difficult to explain. So the other thing is you can do is you can take your iPhone or your non-iPhone, attach a little infrared temperature gauge, which like cooks use, and then you can measure heat. So in mice, brown fat and IWAT, when they're activated, release heat. And you can measure more heat, and that's consistent with increased activation. And you can see here on the right, the actual brown fat dorsal heat activation is higher, consistent with activation of brown fat. And UCP1 in the knockout is higher on the left. And he did a Western blot also. So we have increased energy expenditure, but we have the same body weight. Yes, sir.
That was done with an iPhone infrared sensor.
iPhone with a little infrared device
So you
attached.
just pointed at the mice.
Well, yeah, you don't just point it, but yeah. There's some art and some effort in it, yeah.
Okay.
Yeah, but yeah, this is kind of, we're modern day scientists, my love. Yeah, this is pretty common now. Yeah?
I'm sorry, is this an Amazon purchase? Or is this
I'm sorry?
Is this like something we get freely available? Or is this a special?
Yeah, it costs $300. Read the paper. Like I can send you the paper about it. Yeah, this is pretty standard.
You can start shopping this on Amazon, and we can look at it.
Yeah, but I mean, you can look at the paper. But the thing is, this is so clever. I mean, you know, because it's non-invasive, you don't have to put them in a chamber for days. You just put it, you do it, yeah, and it works. All right. We good? Yeah. All right. I told you that we'd have a diversity of things in this talk today. We're not done yet. Hold on to your hat. All right. So why isn't it thinner? Has increased energy expenditure. So I'm not sure we have a total answer, but we may have a partial answer. Oh, I love the slides. So we know it's not food intake, but what about the gut? I told you about serotonin regulating motility. It decreases motility. Oh, he's got fancy slides. Okay, so we collaborated with Kim Booman, that's her picture, from Purdue. And Kim is one of the nicest people in the world. You know, you have to collaborate with people from the Midwest. They think everything is great. Hi, how you doing? That's like better than Prozac talking to her. She's amazing. And she's really smart. And she's so kind. So she gave us, she collaborated. And she said you can put dye into the food, and then you gavage the mouse, and then you can measure the rate of appearance of the dye in the feces. Like, you know, this is simple, like George Westinghouse. And you can see that the transit time in the upper left is reduced. So the rate at which the food, but that's not surprising. It's many reasons. It's gastric emptying, but also GLP-1 slows intestinal motility. Okay? Don't forget that. I mean, people who take that drug, they complain about this horribly. Well, not horribly, but a number do. And so then we measured the energy in the actual stool using a bomb calorimetry. That's what Kim did. And there was no difference per day, well, in the actual amount. This is the intake, I'm sorry. But there was a different amount of energy in the stool per day, okay? That's what you would predict. Pretty cool. So this is the fecal energy. This is the energy that's lost in the feces. I can't believe he calculated this, my guy. And he showed it was reduced. And so the energy harvest, you know, that's the nutritional lingo, seemed to be higher in the knockout, maybe because of reduced transit time. I have some other ideas now, listening to talks and everything. So that's pretty cool. So we have a adipocyte making, generating a metabolite that circulates in the blood and regulates a number of things. So, of course, we weren't happy. We wanted to know what happens if we did it in the absence of a high-fat diet. So we used a technique that's common that the brain researchers use. And so we generated this AAV, and on the top you can see the AAV has two kind of LOX-P sites, but the cDNA is in the antisense position, okay? And what happens is that in the presence of Cre, it flips into the sense position and gets expressed. The reason we did this is we wanted to express it specifically in gonadal adipocytes. So we took mice that had adiponectin Cre, which is Cre only in adipocytes. We have some surgeons. They opened up the mouse, and they injected this AAV directly into gonadal fat, only one depot, okay? One depot, no, low-fat diet. And then we measured a number of things, okay? I think this is a really cool experiment. I really like this. And indeed, the mouse with the overexpressor had increased body weight, increased fat mass, no difference in lean mass. So it's not the high-fat diet. And the tissue weights were increased, the wild liver, fat weights. Okay. So the important form is that adipocytes are making a hormone that acts distally in the body. I thought that was pretty cool, even as an endocrinologist. And we measured plasma 5-HT. This is just gonadal adipocytes out of higher. Fasting blood glucose was higher, insulin, abnormal GTT, ITT. We did AKT, that was reduced, phospho-AKT response to insulin. So Liver weight was increased. Plasma AST, not so much. What is this? TPH2, this is not in the liver, but increased. This is adipocytes. Average adipocyte size was increased. And these lipogenic genes were increased, and crown-like structures were increased. So this is a very good demonstration of the fact that it can be secreted. That's the key thing. One depot, low-fat diet, and it's getting secreted and having pleiotropic effects. And indeed, we had reduced energy expenditure. Okay? And we did what you like, the thermal imaging, and it was reduced in the brown fat. This just shows that UCP1 was down, consistent with that. And mesenteric fat, TPH2 in the knockout and overexpressor. So the reason we put this in is that mesenteric fat, is pretty close to the intestine, may have some influence. Dr. Fried will correct me, I'm sure. But so TPH2 in the knockout is greatly reduced in the mesenteric fat. The overexpressor doesn't have anything because it's not expressed there. So maybe we don't see any effects on, similar effects on maintaining the body fat. I'm not claiming that. It just may be a partial thing. So I love this. You can't hire these students and pay them enough. You all should get raises. So he added AAV TPH2, and an inhibitor of a specific receptor, and he showed that he could increase AAV TPH2 levels, and the ketamine, which is the inhibitor of the H2R, the receptor didn't have any effect. No effect on TPH1, but the ketamine didn't have any effect on the 5-HT in the media. I love this. It's like, IMAX. So anyway, the AAV increased the expression of fatty acid synthase, DGAT, but when you add the inhibitor, you block the effect. So it's that receptor. That's all. This is gravy. Same thing with brown fat. I don't want to bore you. And the same thing about HT. All right. So is TPH2 only increased in adipocytes of high-fat fed mouse? What about mice that eat chow or overeat it? So the one model is the OB mouse, which just overeats chow and gets obese. So you can see here, TPH2 is greatly increased on the right in OB adipocytes. Okay? So it's not just, and also in brown adipocytes. Look at it in brown adipocytes, it's like, no one studies that, but that's sky high. And the plasma 5-HT is high. Okay? We should probably do that, but OB mice are very hard to make because they're fertile. So what's a common denominator for obesity? It's insulin. So we know great geniuses here. So we added insulin to our adipocytes, and you can see it increased TPH2 expression. And this is protein on the right. And the time course. So insulin, so fasting and feeding can increase serotonin. Because fasting, you reduce insulin, fed, you increase it. These are obese humans, and we found that fasting insulin correlates with TPH2. Okay. So this is the model. You have obesity, insulin increasing. We're not getting into the torque. Increases 5-HT, increases the secretion of serotonin in both mice and humans, and it has peripheral effects, which I think is kind of cool. So these are all the people who helped me. These are the people in my lab who are fantastic. I can't say how great and how nice they are and what a great team they have. John did ACSL5. I didn't put it here, but Brian Park did TPH2. Mike, Kimberly. These are other people. and Reeves. And of course, Susan Freed has helped me very much in my life. So these are the people, NIDA, USDA, Tufts, and the Robert C. Veronica Atkins Foundation. So this is an actual 3T3 cell adipocyte that was differentiated in my lab. And we stained it with perilipin. And after searching thousands of adipocytes, we found the happy fat cell. Now, we haven't decided whether we're going to put it on shirts yet, but we may, and copyright it. All right. So thank you very much. I hope it was good. I can't thank everybody enough for their kindness and support and organization here. Very, very nice. And all these lies about me. Okay.
This is a plaque. It says, in recognition, the Hans Fischer Lectureship, interaction between nutrition and different tissues to regulate diet-induced obesity. Andrew S. Greenberg, MD, April 24th, 2026.
Okay. Thank you so much. I really can't thank you for everything. I thank Dr. Fischer. Now I can show my kids I did something. Okay.
All right. Thank you again. All right. Thank you all for coming today, and have a good rest of Friday and a great weekend. Take care.
Okay. Thank you. Thank you. Thank you so much.